yourself, sir. You can start.
Okay, sir.
Hello.
Am I audible, sir?
Yes, sir. Yes, sir.
Okay. A warm good evening to everyone. It's my honor to welcome all the participants from various esteemed institutions across the country to the AICT Atal sponsored online 6 days faculty development program on sustainable biofuels from Agri and Agro-Industrial waste by Microbes, organized by the Department of Biotechnology at Sri Venkateswara High-Tech Engineering College, Autonomous. Once again, I extend a warm welcome to all the participants for the day 2 FTP. Now we are going to attend the third session of the FTP. It's my privilege to welcome our session speaker of day 2, session 3, uh, Professor Dr. S. Ramalingam Sir from Department of Biotechnology at Anna University, Chennai. With all your permission, I would like to give a short introduction of Dr. S. Ramalingam, sir.
Sir, please, only 2 minutes, a very short introduction, otherwise I'll introduce myself.
Okay, sir.
Yeah, my introduction is not very important here. Yeah.
Okay, sir is a investigator, is a chemical engineer turned biotechnologist specialized in formation of genetically manipulated systems in bacteria. The investigator has a well-structured industrial career spanning 5 years in three giant pharmaceutical companies. He is offering bioprocess and metabolic engineering courses for MTech, BTech, and PhD students at Anna University, Chennai. Further, he has completed 10 funded projects, including one international project. He has registered 6 patents in his domain and published more than 25 articles in his trusted areas. We are very happy to have him as a session speaker today. With all my respect, I invite Professor Dr. S. Ramalingam sir to deliver his lecture under the topic of enzymes formulation for treatment of ecological biomass. We welcome you, sir. The
Yeah.
session is over to you. We are very eager to hear your words, sir. We welcome you, sir.
Thank you very much, Vigneshwaran, Dr. Vigneshwaran, for your kind introduction. And at the outset, I thank Ramalingam, Professor Ramalingam, and Dr. Tangavel, the principal of the college, for providing me this opportunity to deliver a lecture in this particular series on ethanol making. So I'll basically talk about how we try to produce 2G ethanol. Okay, second generation ethanol. There are some 70, I hope some 79 people are there, 79 faculty members are there. You can freely interact with me, stop at any time, ask questions. It's absolutely fine with me. And it's more of a dialogue rather than merely displaying the lecture. But what we have developed here at Anna University is that we developed the enzyme, cellulase enzyme system, and then we transfer the technology to Hindustan Petroleum Corporation Limited in Bangalore R&D, and they have got the strain, they have got the process, and we're going to jointly scale it up, and it will be commercialized. So I'm here today to tell you what it takes to develop a technology from the academics and translate it to the industry. And here we have developed this cellulase enzyme process for 2G ethanol. With this brief introduction, I'll go into the presentation. Can you all see the slides? Sorry, just. Can you, Ramaswamy sir, can you put the slides?
Yes, sir.
Yeah, you just open the slide. I'll make an alternate.
Okay, sir. Okay, sir.
Are you putting it?
Yes, sir. I'm.
Okay, okay.
Sir.
Yeah, yeah. So, what are the? Please kindly mute it, sir. Sir.
Yes, sir. Yes, sir.
Yeah, mute it. Yeah. Okay. So, what are all the challenges associated with 2G ethanol production in India? So, I am Subramaniam Ramalingam, from Department of Biotechnology, Anna University, Chennai. Next slide. Yeah. So, first slide. Next slide. Yeah. So, currently, we are importing huge amount of. I'm sorry. We are importing huge amount of crude, one of the major importers of the crude in the world, and we imported around 185 million metric tons of crude to the tune of around $500 billion. And when we can blend this petrol with 10 to 20% of ethanol, then we can save a foreign exchange of around 24,000 crore. So, which is what is we are able to see here. And so currently we are blending around 15%, and we want to blend 20% of the petrol with ethanol, which means one-fifth of the crude import can be reduced. And we are currently making around 700 crore liter of ethanol from molasses and from corn. 50% is from corn based and other grains, all the grains, including corn, and other 50% is from the molasses. And the problem with this particular 1G ethanol is that the amount of water usage is very high, and also we are trying to encroach upon the food production and you divert the food for fuel purpose. So food versus fuel is a major concern when we are trying to produce the first generation ethanol. But the only way we can solve this problem is that when we can use the biomass, lignocellulosic biomass, or the municipal waste which is generated on this planet, which can, in fact, completely replace the entire petrol demand if you can convert it into ethanol or any other high density alcohol. But so the PPT has disappeared. Hello. Hello. So PPT is not there.
Sir, just 1 minute, sir. Just 1 minute, sir.
Yeah. So we can completely replace.
Yes, sir. Now it's visible, sir.
Yeah, you mute your mic. Yeah. Yeah. So we can completely replace the entire petroleum requirement with ethanol or any other biofuel. But the major challenge is that to make a cost-effective conversion of this cellulosic biomass into sugars and then convert the sugars into ethanol is a major challenge. The reason being that plants, they have evolved over the millions of years, and no bacterial systems are capable of hydrolyzing this plant material. And if it is able to do that easily, then no life can prevail on earth. So it's very hard to convert this plant material into sugars, and enzymes are able to do it under anaerobic conditions and not at the rate and the scale at which we are really in need of, so that we can commercially, viable manner we can produce ethanol. So that's the major challenge. But nevertheless, across the world, so much of research is being done to figure out how to efficiently convert this plant biomass into biofuel. And one of the major challenges we face, the first challenge we face is that we need to first of all pre-treat this biomass so that it will be amenable for the enzymes to work. And it is a complex array of enzyme systems which is required to break down this complex cellular material into sugars. That's the major problem. But another major problem we are facing is that it's not only that we want to produce ethanol from this cellulosic biomass, but unfortunately, in our country and few of the South Asian countries, we are burning the plant, especially the paddy crops after the no, after they harvest it, the other crops are harvested, they try to burn the paddy husk or rather the straw, the paddy straw, rice straw that is left out is allowed to burn, and it creates a huge pollution problem. And the pollution problem is to the tune that people suffer from various ailments, and we can't really gauge what is the level of financial losses we incur, apart from the fact that it creates a human great no misery to human lives. So India decided to produce this, convert this paddy, no straw, into ethanol, even if it is going to be costing more than 100 rupees, 150 rupees it's costing. They have decided to produce this to make 1 lakh kiloliters of ethanol per day. The investment cost is 100 crore for 1G ethanol, while to convert this agro waste into sugars, we require 1,000 crore. But nevertheless, we are trying to set up 12 2G second generation ethanol plants at an investment of around 14,000 crore. So government has desired to invest, and already in Panipat and few other places, this 2G ethanol plant has commenced. They have started installation, and even their trials are being taken up. And soon we will start making in all these plants 2G ethanol. Next slide. Yeah. So this is the photograph of cellulosic ethanol plant, wherein the biomass will be converted into sugars, and then the sugars will be made into ethanol. Next slide, please. Yeah. So what are all the challenges with this second generation biofuel? First of all, you need a very high energy intensive pre-treatment process. The plant material cannot be directly be converted to sugars that easily by enzymes. So we need to have a pre-treatment. That's the first major issue. And the cost of cellulosic ethanol is higher than that of the other petrol and even corn-based ethanol. That's the next challenge we are facing. I said in India we are able to overcome it because we don't mind spending more money because its medical problem is involved. And the other problem is that investment cost, huge investment cost has to be made. Only then it needs to produce at a very large scale. Only then the cost of the product ethanol can be brought down. And in US, they're able to show that this 2G ethanol can be made cost effectively. And there are a couple of plants. There are a lot of plants which are in the pilot scale level. The economics is getting revealed now. But we are in a position that we're going to go ahead with this 2G ethanol. So we can all start working with 2G ethanol process. Anything related to 2G ethanol, that will be a welcoming thing in our country. Next slide. Yeah. So what are all the major aspects? As I told you, the first is the pre-treatment, biomass. So that's the first step. And the second step is converting the cellulose and into sugars. So that we use enzymes for this particular purpose. And it's a cocktail of enzymes. It's a cellulose enzyme system, we call it as. And then the next step is to convert this sugars that are produced by this hydrolysis, both pentoses and hexoses, efficiently, and convert them into ethanol. And the fourth step will be to carry out the ethanol recovery, fermentation followed by recovery of ethanol. So these are the major steps involved in trying to produce second generation ethanol. And the major composition of any biomass, if you take, it has three major components. One is cellulose, roughly 30 to 40 percentage. You can say one third of it will be cellulose, and the cellulose can be completely be converted to glucose, hydrolyzed into monomers glucose. The other component, which is roughly 30, also one third, is hemicellulose. Hemicellulose, if you try to hydrolyze them, you will get an array of sugars, both pentoses, mainly xylose and other sugars. And therefore, the organism has to efficiently utilize all of these sugars. Only then we can have a very high yield of ethanol from the sugars hydrolyzed. So that's the next major challenge. And of course, ethanol has to be distilled, and it is hygroscopic in nature. It consumes a lot of water. That's another major challenge. But nevertheless, we are producing ethanol by second generation. So the major drawback in this particular 2G process here in our country as of now is the enzyme coptane. Next slide. Yeah. So the same what I've described, cellulose, hemicellulose, and yeah, the third component is the lignin. Lignin is more of aromatic in nature. And we can make a lot of other compounds, useful compounds from this lignin. So it's a valuable compound. So the cellulose and hemicellulose have to be efficiently converted into sugars. So that's where the challenge lies. And that's where we need the enzymes as well. Next slide. Yeah. So there are various kinds of pre-treatment which will make the biomass amenable for enzymes to be treated. And yeah, slide. Yeah, slide. Yeah, one of the major widely used physical treatment process is steam explosion, and but in India we are primarily using alkali-based chemical treatment. So there are acid-based, ozone, and other organic solvents also. But primarily we are focusing on Industrial Petroleum Corporation Limited, IOCL, and other companies. They are working with biomass treated with alkali. And the major biomass source is actually the paddy straw. That's the major biomass. And municipal waste also we can use. Bagasse can also be another major biomass which we can use. Next slide. Yeah. So this enzyme is a cellulase enzyme. It's a widely used enzyme. It's used in paper industries, textile industries, and several other industries. And it has a huge market demand, global market demand of greater than 2,000 million US dollars worth turnover is there. And the pharma giants, along with these manufacturers, international giants, they produce these enzymes, and the major producers are Novozymes from Denmark, and AB Enzymes, DSM from Netherlands, Codexis, Genenca, and DuPont Amino Enzymes, and many other Chinese companies are also producing these enzymes. So they get very high titer, and the enzyme actually it is a group of enzymes which does this job, primarily consisting of exogluconase, endogluconase, and beta glucosidase. Apart from that, there are tens of other enzymes which act synergistically to degrade the biomass, and we usually measure this enzyme activity in terms of filter paper units. So these filter paper units are nothing but we try to have a filter paper which is made of cellulosic material. And we add a certain amount of enzyme and allow this filter paper material to get hydrolyzed into glucose and other sugars, and we measure the amount of glucose that is released by a certain amount of enzyme, and that is how it is called filter paper units per ml. We try to quantify them. And ideally, for you to have a commercially viable enzyme production, you need to have around 10 filter paper units per ml. That is the level of enzyme activity we require for it to be commercially viable. I'll go to the next slide, subsequent slides, I'll show you how to calculate the cost. So here in our lab in Anna University, this project was funded by Department of Biotechnology under Virtual Enzyme Center, IIT Madras, IIT Bombay, Delhi University, Indian Oil Corporation, and Anna University and Jawaharlal Nehru University. All these institutes were jointly funded to the tune of around 5 crores several years back by the government of India to develop this enzyme system so that it can be indigenously produced. And in that process, the Professor K.J. Mukherjee from now currently is working at IIT Delhi. Earlier he was working in Jawaharlal Nehru University. He provided the strain, high producing strain, and we at Anna University developed the strain, the process, scaled up the process, and we got a filter paper unit between 8 and 9 filter paper unit in 55 to 60 hours. So, but the commercial viability is around 10 filter paper units per ml and above. And therefore, this technology was taken up by HPCL. And the operating cost, we shall calculate only when you go above 10 FPU per ml, then it becomes a viable titer. Next slide, please. Yeah, so the time is also very important, and most of the cellulase producing fungus, usually it takes a fermentation time of around 1 week, at least 4 to 7 days. They usually even more than that, 10 days. But operating the fungal fermentation is expensive because you need very large reactors, and it's highly viscous, and oxygen transfer is a very big challenge. So the power consumption will be 4 to 5 kilowatts per meter cubed of the broth and per hour, and therefore agitation cost will be huge, and most of this fungus will be growing efficiently at 25 degrees in that range, and you require chilled water. That's also a very expensive commodity to maintain the temperature. So agitation and chilled water and also aeration, all of these things makes this process expensive. So trying to minimize this time is very crucial. So we have worked on that, and the basic idea is that in the first 20 hours or 40 hours, you can see we have to rapidly build the biomass. So that's the very important thing. And usually we quantify the amount of biomass in the fungal system by a method called packed cell volume. So the packed cell volume is nothing but in a total volume, when you centrifuge it at a certain G force, what is the amount of biomass, the volume it is occupying. So that's called packed cell volume. Roughly it should be 40%, 40 to 50%. Ours is little between 50 to 60%. It is slightly overgrowth. Then it becomes very viscous and oxygen transfer will be an issue. So the initial amount of glucose or any carbon source you take should rapidly allow this biomass to build up. Within 24 hours, preferably 20 to 30 hours, you should be able to get the 40 packed cell volume of biomass, fungal biomass. It will also have a lot of other solids in them, cottonseed flour, soybean flour. We add microcrystalline cellulose. All of it contributes to the volume also to some extent. So once this biomass is built up, then the rapidly utilizable carbon source and even the nitrogen should get exhausted fully, and then we have to feed them. So during the stationary phase, in the batch cultivation process, it produces the enzyme during the stationary phase, or in a fed batch process, in a slow growing phase, you have to make the fungus to grow very slowly for the next 30 or 40 hours and make them produce the enzyme. Same like antibiotic fermentation, whether it is penicillin, streptomycin, cephalosporin, all of these antibiotic producing fungal strains also we just do it in the same way. So I have worked in the pharmaceutical industries and I'm a specialist in fungal fermentation. So I've done work at the lab scale and to the largest scale, 200 meter cube reactor I've operated, one of the largest in the whole of this continent, Asia. And we need to scale up the process. All of these antibiotic fermentations, we just do the same way, fed batch. So initially, rapidly, we have to make a batch process and grow the biomass, and then allow the biomass to build up very slowly, close to the stationary phase, but still they will be viable and they'll keep producing this antibiotic and enzymes. The enzymes are also produced in the non-growing phase, and we also tried to provide feeding. We tried lactose and several other carbon source also. Glucose, we get catabolic repression. That's one of the major drawbacks in this wild type fungal strains. So the glucose concentration needs to be kept very low when the enzyme is about to be produced, because the very idea of the organism to produce this enzyme is to hydrolyze the cellulose into sugars. And when sugars are in high amount, there's no need for the organism to produce the enzyme. So they repress the enzyme production. So that's the major challenge. So we are able to do that, and we successfully developed a batch process and also fed batch process. But I'm not able to show you the fed batch process with increased efficiency here. But right now, we are able to show that even batch process can produce considerable amount of cellulose enzyme, which is very close to commercial viability. So the next slide. Yeah. So roughly 1/3 is the cost of making this sugars from this pre-treated biomass. So enzyme systems are very important, and usually we make one fermentation to produce all the three enzymes I've mentioned, but they may not be in the optimal ratio, but can formulate them. For example, beta glucosidase will be the rate limiting step in all these systems. So we can add beta glucosidase separately, and there are organisms which produces this enzyme and then formulate the appropriate ratio so that there will be maximum conversion of biomass into sugars. Next slide, please. Yeah. So for all this, for translating any process to the industry, the most important thing is the economics. What I found is that academicians across India, they do so much of work, but they are not sure where their process is at, on par with the industrial process. So whether they are close to viability, whether they are far away from viability, nobody is aware of it in the very first place. So that is a major bottleneck I find when people are trying to translate their work to the industry. So the best way to do that is to calculate the operating cost for any process you are working. And if you take, when you consider the operating cost, what you need to consider is that for you take 1 meter cube of a fermenter vessel, and you take one batch process for that, and how much of medium components you're going to add, and the medium components you have to calculate the cost of these components at the industrial scale. For example, they're going to get even we are using glucose as the carbon source. Glucose is around 0.35 rupees, 25 to 35 rupees per kilogram. So little more than that also may be there, but you should be able to gauge that cost. So it's around $0.5 or $0.3, $0.4 per kg will be the cost of the carbon source. Likewise, every raw material, whether it's salts, whether it is soybean flour, so all of these costs have to be calculated. And that's very important. For the sake of preserving the confidentiality, I'm not showing you the medium composition as such, but the costing has to be done like that. And once you calculate that total cost, then the utility cost has to be calculated. So how much of steam you are required for sterilization, and what is the amount of cooling water? What's the price of cooling water? Then you may have to when you're going to bring the temperature after sterilization medium from 121 to say 45 degrees, you can use cooling water. The cooling water is made by cooling tower. It's cheaper. But from 45 to 25 degrees centigrade, you have to use chilled water. That's very expensive. 7 to 14 degrees chilled water will be used. So the cost of chilled water also we should know how much of chilled water we require, and how much of cost it is incurred, and what is the cost fraction in that utilities. So you can see agitation cost is one third, around 40%. Chilled water cost is 30 percentage, and compressed air is around 20 percentage, one fifth of the total cost. So that comes around $38, roughly $40, and the medium cost is around $60. It can be made into, they can cheaply make it. Yeast extract is an expensive component. Microcrystalline cellulose, they're all the expensive components which can be cheaply made when they are produced at large scale. So that will another cost another $30 to $40. So $70 to $80 will be required to make 1 meter cubed of fermentation broth. And therefore, in that fermentation broth, you should have $100, minimum $100 worth material product. So that's how this calculation spans out. So if you're making 88 FPU, that means 8 to the power 6 filter paper units per meter cube. Because I have calculated 8 FPU per ml, that works around 8 to 10 to the power 6 FPU per meter cube. You can have a costing of around $80. So the cost, what is the cost of the enzyme that is sold in the market? That's very important. How much of enzyme I'm making, I told you. And one of the major cost sources available is that there are three methods by which you can find out. One is that you can talk to the industry people who are working in the same area, and they can tell you the cost of the product. That's the first way, cost of the substrate, everything you can ascertain. But that's very difficult. We have to get access to the industry. This next method of assessing is that you can go to any bulk site, bulk buy products sold in bulk like India Mart and Alibaba in China. That's another source from where you can find out what is the bulk cost of cellulase or any other enzymes or any other product. So as you can see, roughly it costs around $100, around anywhere between $40 to $130 per kilogram of enzyme, and that 1 kilogram will have 10 to the power 10 into 10 to the power 6 FPU filter paper units it will have, and that's the cost of the cellulase. But Novozymes and other companies, giant pharma companies who are making these enzymes, they are pricing it much higher. And when you want to produce it on a very large scale, you need to have a very reliable supplier in the form of Novozymes and other producers, DSM and all. And it's expensive. Enzymes are expensive. So it's cost 2 to 3-fold more than what I am quoting. But we should know what exactly it takes to produce this enzyme at the largest scale. And other than that, what is the cost of the enzyme? These two things you need to know. Next slide. Yeah. So we have initially started working with wild type. And the first thing we may have to do is to remove this catabolic repression. Even when you feed glucose, even in the presence of glucose, enzymes should not get repressed, enzyme production. Only then we can do fed batch. So we'll continuously keep feeding glucose and extend this productivity phase. We initially in the batch you have only for 30 to 40 hours or even less than that, but we need to extend it to even 50 to 60 hours. Then the product should get continuously produced, giving us high filter paper units. So currently we are focusing on gene manipulation and also process optimization. And the genetic manipulation is done by IIT Delhi by Professor K.J. Mukherjee in our product. And we will reach a target of more than 10 FPU. That's where we are focusing upon. And the process also will be scaled up by the company, Industrial Petroleum Corporation Limited, oil corporations. They have the pilot plant, and I went and saw their 2G pilot plant. They have a beautiful second generation plant to, you know, to take trials and also to develop processes. Next slide. Yeah. So there are a lot of targets apart from the fact that the catabolic depression has to be overcome. Scientists in the last couple of years they have identified tens and tens of gene manipulation targets. And the major problem, challenge with this trichoderma system which we are working is that gene manipulation tools, availability of gene manipulation tools, even transforming the plasmids and bacterial systems are relatively easier. Fungal systems are extremely tough when compared to them. So we need to develop vector tool sets for any particular strain or if this vector tool set is available for a strain, then we may have to choose the targets and start doing gene manipulation. Yeah, next slide. Yeah, the same slide. There are various methods by which we can do the gene manipulation. CRISPR-Cas is available right now, which will enable us to rapidly go for targets. And that's how we are planning to take it up. Next slide. Yeah, so the various targets I'm showing, just a sample of targets we are showing. We'll identify those targets and knock out, overexpress these targets and regulatory aspects, and then we'll try to enhance the enzyme production level. Next slide. Yeah, so we also try to Saranya Ramar. Okay, we also try to immobilize this enzyme. One of the major challenges is that a lot of solids will be there. So we try to use CLIA, cross-linked enzyme aggregate, with a magnetic CLIA with the help of magnetic ion. With the help of ion, we wanted to form this enzyme immobilization method so that we can put a magnet and separate these enzymes after it is subjected to hydrolysis of the biomass. So we try doing them and we are able to extend the cycles, but the enzyme is not very stable after 5 cycles, but still it's a good method, and we also try to do that in our lab. Next slide. Yeah, so this is our team, and Dr. Prabhakaran Krishnamurthy is the consultant for us, and currently he's in Hindustan Petroleum Corporation Limited. He's working as subject matter expert, and we developed, my students Revati and Rokit and Akash, they developed this process with the help of Professor K.J. Mukherjee from IIT Delhi. And next slide. Yeah, so this is the technology that we transferred to Hindustan Petroleum Corporation Limited, Bangalore, by our Vice Chancellor, is the former Vice Chancellor, Registrar and Dean. And on the other side, we have the esteemed members from the Hindustan Petroleum Corporation Limited. Next slide. Yeah, so once you make these enzymes cost effectively, the next major challenge is that we need to hydrolyze these. After the sugars are made, the organism has to efficiently utilize both glucose and xylose. That's the major challenge. Next slide. Yeah, one of the major problem with this ethanol producer is that Saccharomyces cerevisiae and Zymomonas mobilis, these two organisms are the best ethanol producers available to us. They produce a very high ethanol concentration of around 100 grams, greater than 120, 130 grams per liter. And they can also tolerate, currently they are able to tolerate 130, 140 grams per liter. And the volumetric productivity should be greater than 1 gram per liter per hour. Currently it is around 1 to 2 gram per liter per hour. And they should be able to grow in a very simple medium. They should be able to resist inhibitors. and low pH, high temperature. In all these cases, they should be able to survive and produce high concentration of ethanol. We found only these two best producers. Unfortunately, the problem is that both these organisms, they can utilize glucose efficiently, but they cannot utilize xylose. Next slide. Yeah. So as you can see in this slide, glucose is converted to pyruvate, and from pyruvate, we can make ethanol. So the NAD/NADH will be recycling between the EMP pathway and between acetaldehyde and ethanol. So redox is balanced. That's how ethanol is produced. But they are not capable of utilizing xylose. Next slide, please. Yeah. So in order for these organisms to utilize xylose, we need to efficiently engineer xylose transporter into these organisms. They have done that. And they cloned three enzymes: xylose reductase, xylitol dehydrogenase, xylose kinase. So three enzymes which will convert xylose to xylose 5-phosphate. And xylose 5-phosphate is one of the intermediates in the pentose phosphate pathway, as you can see in the diagram here. And the trans-ketolase and trans-aldolase enzyme, the pentose phosphate pathway, also have to be engineered so that high amount of xylose can flow back into the glycolysis pathway to make pyruvate, from where ethanol can be made. And they have successfully engineered. Scientists across the world have engineered both Saccharomyces cerevisiae and Xanthomonas nobilis to efficiently utilize both xylose and glucose. But right now in India, we don't have a strain, and the scientists are working, a couple of institutes are working on them. And if you can develop such a strain, it is a very valuable thing. And currently we are getting it from abroad. Next slide, please. Yeah, so numerous hurdles you will face when you try to clone all these enzymes. The first step is that the first enzyme requires NADPH, the second enzyme requires NAD. You will get a redox imbalance. To solve this redox imbalance, they try to modify the first enzyme such that it can use NADH instead of NADPH, so that NADH will become NAD. The second step, NAD will become NADH, so that the redox will get balanced. So they did all this exercise, and they also figured out a bacterial enzyme which can be successfully expressed in Saccharomyces cerevisiae from a Pyromyces species, and then they are able to solve this problem. Otherwise, it started making xylitol rather than getting converted to ethanol. So initially, scientists faced these problems. Right now, they're able to overcome, and now they're able to efficiently produce ethanol from xylose. Next slide, please. Yeah, so this slide is about from Xanthomonas nobilis. Also, they engineered the entire organism to efficiently make ethanol from glucose and xylose. Next slide. Yeah, so E. coli, recombinant E. coli is a very good utilizer of sugars. It can utilize glucose, xylose, and a lot of other sugars very efficiently, but they produce very less amount of ethanol. So scientists have cloned pyruvate decarboxylase and alcohol dehydrogenase gene into the organism, and you can see that they're able to produce high amounts of ethanol, but not as high as Saccharomyces cerevisiae, but nevertheless still recombinant E. coli produce a very good amount of ethanol. Yeah, next slide. Yeah, so they're also trying to use other organisms like Klebsiella oxytaka and even thermophilic organisms, where they try to produce ethanol. So we can operate the fermentation at a higher temperature so that ethanol can simultaneously be removed, or even a continuous process can be developed. So if you apply a vacuum, then ethanol can be continuously removed so that product inhibition will not be there. Ethanol is a very toxic compound. So scientists are trying to do that also, and they're also trying to use a plant pathogenic organism, trying to they'll produce a very sophisticated system, enzyme systems, so that the same organism will be made to produce ethanol. So we can dump biomass into the reactor so that we can simultaneously convert this plant into sugars and also sugar into ethanol. So such work is also being taken up, and is at the research stage level. Next slide. Yeah, this is the thermophile, the Thermoanerobacterium saccharolyticum, which try to produce ethanol. And it's a very welcoming thing because contamination problem will not be there, and only cooling water is sufficient to maintain the temperature. We can simultaneously remove ethanol. So for all these reasons, they are trying to work with thermophiles. Next slide, please. Yeah, they're also trying to produce it from algae, from blue-green algae and from algae. Currently, they are trying to produce biodiesel and also ethanol, but it's at a very low titer levels. Next slide. Yeah. So, thank you very much for your patient listening. And these organisms are all being continuously developed, metabolically engineered organisms, so that once the hydrolysis of the biomass is done very cost effectively, we will be able to see more and more number of plants across the world trying to produce second generation ethanol, which is a carbon neutral process. So global warming is a very catastrophic process. We know that right now we're able to see a very intense rainfall and drought and flood. We're able to see that the only way to mitigate this problem is to replace all the biofuel requirement in a sustainable manner, at least a carbon neutral process where the carbon dioxide will be consumed by plants and plants will be again converted to fuel and be burnt, and the carbon dioxide will be put back into the atmosphere, which means we don't add any new carbon dioxide levels to the existing concentration. So in that regard, this 2G ethanol is a very welcoming process, but then it's a very challenging, huge investment cost is also required. But nevertheless, I'm sure that the next couple of years, the humanity will try to harness this 2G ethanol in a very big way. Thank you. So the forum is open for discussion.
Dr.
Yes.
Ramalingam, I have a
Yeah.
question. There is a lot of controversy regarding the use of corn for ethanol.
Yes.
And what I have seen in recent studies, as late as 2022 and 23, is that there has been more emission from the use of ethanol in America, greater than even gasoline.
Yeah.
What do you say to that?
Yes, it's a major issue. So in order to they it's a they have to sustain the farming practices. They need to encourage their farmers, and they need to minimize the import of crude from other countries. And for these reasons, we are trying to produce ethanol from corn. But definitely, yes, we use so much of water, and if you do the entire life cycle assessment, uh, the input we are providing is not a very sustainable operation from corn.
So, are we in India promoting it in any way? Because I see some reports as well
Yeah,
that, especially in northern India, we are trying to grow more corn. Are we
yeah.
trying to ape the US without understanding the repercussions?
Not the repercussions, madam. We need to sustain the farming, and we need to provide. We need to make ethanol. No other way. But a better way of making ethanol or any biofuel. So the more and more unconventional energy sources available, we can replace them. That's the best way possible. The wind energy, for example, if you generate electricity out of it, if you're going to run a transport out of it, it's much better than that. But
Right.
all said and done, we are trying to do better and better, whatever is possible. But how much of water we use, how much of other energy sources you are using to grow the plants and then produce ethanol out of it, all that needs to be considered here.
And also the effect on other food growth, right? Because
Ye-
agricultural land taken up for growing corn, for example, could be
Yeah.
used for other crops which are more beneficial to Indian society, right?
Yes, yes, madam. That's also more than Indian society. A lot of African nations, for example, they don't have food actually. So it's a major problem, and we need to minimize them. That's why we prefer 2G ethanol from biomass, non-edible biomass. We try to produce sugars from them and then try to produce ethanol out of them. That's the best option actually. So we can use arable lands and arid lands which are not suitable for cultivation. In such
Okay,
cases, if you're going to use them, that will be much more beneficial.
okay. Thank you.
Yes. You are from which place, ma'am? You can introduce yourself.
I am actually from Coimbatore. I did my BSc in Avnashalingam, but
Okay.
subsequently I did my MBA from Bharatiyar University. But
Okay.
presently I am a professor of marketing in assistant professor in UPS in Dehradun.
Fantastic, ma'am. Fantastic. Yeah. Any other questions? Feel free to talk. We'll discuss what's anything you can discuss actually.
Participants, you have any questions?
Sir, I like.
Yeah, Ashima
Yes, sir.
Katuria.
Yes,
Yeah.
sir. Yes, sir. Sir, I like to ask, in one of the slides you have shown that cellulosic ethanol is costlier than corn ethanol. Why is it so?
Well, because corn can easily be hydrolyzed. Okay, amylase. If you add amylase to corn, it will get converted sugars. But to convert the cellulosic biomass into sugars, you need steam explosion or alkali hydrolysis. The huge amount of this,
It is a complex carbohydrate whereas corn is purely gluten.
yeah,
Okay.
very simple. So the investment cost is 1,000 crore to make 1 lakh kiloliters per of ethanol per day. The investment cost is 100 crore for 1G ethanol, 1,000 crore for 2G ethanol. So when you are going to put 1,000 crore into it, the cost will be there. The
But the
fixed
major
cost.
issue is the utilization of beneficial use of the biomass. So there
Yes.
we should approach cellulosic only.
Yeah.
Ah,
Yeah. So you can introduce yourself, madam. You are from which place?
Uttar Pradesh. I am from Uttar Pradesh,
Okay.
Meerut Institute of Engineering and Technology, professor in Department of Biotechnology.
Very good. Very good.
I also work in production of enzymes only.
Very good.
Yes.
Very good. Yeah, Nivedita, Miss Nivedita Divedi.
Hello.
Hello. Yes, madam.
Yes, sir.
Yes, ma'am.
Actually, sir, I am on the way towards my home. So,
Yeah. So you have any questions, madam?
right now, no question, sir.
Okay. Okay. I saw your photograph. That's why I asked. Yeah. Any other questions?
Participants, any questions? Anything you want to interact with a speaker?
Brasna Kandaswamy has raised a hand. Yeah.
Prasno Kandasamy, sir.
Yeah. You introduce, sir, and then you can ask questions, sir.
I am from Tamil Nadu, sir. I am working as an assistant professor in Electrical Engineering College. It is in Namakkal.
Okay.
Sir, actually, we have a land in Dharmaburi, sir. It is a dry land one. Can you tell me any other crop for this dry land area? We have
No,
nearly.
sir. Yeah, I'm not an agricultural scientist. Probably I'm not the right person to advise you or give inputs. You can take it from TNAU straight away from them. You go figure out for soil, what kind of crops can be grown, and you want to convert that biomass into any other valuable products.
Definitely. Thank you.
Yeah.
Dear participants, any questions? Any interactions?
So how many of you are developing technology for the industries? How many of you are there? Are you working on it? Anybody is seriously working on it? Yes.
I think no more questions. I think so.
Okay.
Now I request Miss Aswini ma'am, Assistant Professor, to deliver the summary note of expert talk.
Before that, I thank the organizers for providing me this opportunity. And Ramalingam, Professor Ramalingam, the principal, Dr. Tangavel, and the coordinator of this program. So thank you so much, and we can look forward to interact again. Thank you.
Thank you, sir.
A special thanks to our speaker, Dr. Ramalingam sir, for sharing his expertise and providing us with such valuable knowledge. Your presentation was truly insightful and inspiring. Thank you, sir. The concise and summary of the session is that while 2G ethanol has potential for reducing the dependence on fossil fuel and enhancing sustainability, challenges in feedstock supply, technology, economics, and policies must be addressed. Strengthening R&D, improving government incentives, and developing an efficient supply chain are crucial for the success of 2G ethanol production in India. And that is the precise of this session. And thank you one and all for attending this session. Thank you, sir, and thank you all the participants.
Professor Saladev ma'am.
Yes, sir.
Ma'am, you can.
Good evening to everyone. It's my pleasure to introduce our esteemed resource person for today's faculty development program, Mr. K. Sanmugam, sir, Senior General Manager at Svhec Paper and Boards Limited, Pallipalayam, Erode. Mr. Sanmugam, sir, is a distinguished professional with over 5 decades of experience in water and wastewater treatment. He holds a degree in chemistry from the University of Madras in 1970 and a postgraduate diploma in industrial pollution control from Annamalai University. Since joining Svhec Paper in 1971, he has contributed significantly to the field of water conservation and effluent treatment. His key achievements include implementing innovative water conservation processes to reduce water consumption in paper manufacturing, establishing an anaerobic lagoon for high CO2 effluent treatment, and utilizing biogas as a fuel alternative in lime kilns, developing a lift irrigation system using treated effluent for sustainable sugarcane cultivation, advocating circular economy practices such as using bagasse from the sugar in paper production. He has also published several research papers in IPPCA and has served as management representative for ISO 14001 implementation. With his vast experience and pioneer work in environmental sustainability, we are privileged to have him with us today. I request our well-regarded resource person to deliver his presentation. Sir, please. Hello, sir.
Ma'am, I think Sir left. Yeah, Sir came, ma'am. Sir came. Sanmugam, sir.
Shanmugam sir. Sir,
Shanmugam sir. Sir, I think you are muted, sir. Shanmugam sir.
Hello.
Yes, sir.
Yeah. Good evening, everybody.
Good evening, sir.
Yeah. I'm basically Shanmugam Seshan. I would like to share with you with my own experience in the areas of wastewater treatment as well as in other related areas in pulp and paper industry. It will be much more useful to the participants. I hope so. Basically, this paper industry, it was started in 1960. Some preliminary, this one I will tell you about the industry. This was started in the banks of River Kaveri somewhere in 1962 for a production of hardly about 25,000 tons per annum. As the first paper industry in Tamil Nadu installed in the regime of Kamraj and Mr. R. Venkatraman and all. Then they have started that production increased to 35,000, then followed with 55,000, now 1,65,000 tons per annum. And by the way, they had one more second unit in Tirunelveli also, that also producing a valuable export variety paper to US market and all, right? Basically, everybody is aware that paper industry is one of the vast consumer of water. Earlier, when I joined the mill, we have consumed around somewhere around 450 cubic meter per ton of paper production. Now we are in the range of hardly about 35 to 38 cubic meter per ton of paper. Based on the new technologies, by the way, various innovative measures taken inside the mill, all these things are helped in reduction of water. This is one part. Number two, in those years, this extra water, whatever we produce, it is supposed to go to the river, polluting the river and creating a lot of unwanted issues in the general public. Then we have selected an innovative irrigation scheme suitable for growing sugarcane, other tree crops also. By the way, we installed a sugar industry named as Pony Sugars for production of bagasse. It is a major raw material for us. And incidentally, sugar is a byproduct for them. This is the story of our industry. And coming back to that, our wastewater has to be used by the farming community without getting into the detrimental effect to the soil and etc. productivity also. In that angle, we have done segregation of wastewater at source and whatever be the high COD, high BOD effluent and all segregated. In that angle, how to go about with the biological treatment system, thereby reducing water consumption, thereby reducing energy, thereby reducing the carbon footprint also, right? In that angle, we segregated bagasse effluent. Okay, bagasse is the raw material received from our sugar industry, which will be initially slushed. The residual sugar, what is contributing for high COD and BOD, is taken to a tank where it is slushed. Bagasse is taken back to the paper making process. The wastewater containing residual sugars with a COD of 10,000, BOD of somewhere 3,000 to 3,500 is taken to the anaerobic system. Suppose if the system is not there, I have to use much more aerators, run with huge horsepower and all. This is the negative aspect in that. That a biological process taken care of into the anaerobic system. We constructed an anaerobic lagoon in collaboration with our team. Then we took that entire water, about 1,000 cubic meter with a COD of 10,000, goes into the tank where it is subject to treatment with the first is, I will go through that other process related issues. Then I will be in a position to continuously, I will get about 90% reduction across the anaerobic system. That is reduction in COD as well as BOD also. Incidentally, earlier, this methane was being outside, it is all going to the atmosphere. Then we thought of going for collection. Why don't we collect that the biogas and use it in the lime kiln, where it can replace the furnace oil. Then we located on a Sriram fiber fellow. We got that balloon. We covered that anaerobic system, collect the gas, and pump it over to a rotary lime kiln, where I reduced the lime kiln oil consumption by about 4 kiloliters, which is equal to somewhere around 2 lakh rupees a day. That is a huge potential, in addition to saving of our good amount of money for the company. This is the beginning story. Let me proceed with that treatment process. Basically, as I was telling you, the water quality was 3.5 to 4 pH. Incidentally, it will be very low pH because of that volatile fatty acids and all, like acetic acid, etc. Then that has to be increased with the pH by addition of lime as well as little caustic also, to reduce the pH to 6.5 to 7.5. Then the 10,000 COD or the BOD 3000 across the lagoon with a 10 days retention time, I am able to get a 90% reduction across the lagoon. Thereby, only about 250 to 300 ppm only will be the BOD, and my volatile fatty acid has reduced to 550. The alkalinity will be 1500. Good amount of biogas will generated when the ratio VAB to alkalinity ratio, particularly, that is still the key factor actually. It should be maintained between 0.3 to 0.5, so that your biogas content, that is methane content, will be 65 plus all the time. That is the beneficial aspect by keeping this one. If you go through the anaerobic process, it is a universal only. The organic matter, carbohydrates or proteins or fats or lipids, hydrolysis, it will undergo a hydrolysis. The soluble organic molecules, sugar or amino acids or fatty acids, are converted by volatile fatty acid. It is called as acetogenesis, fermentation. Can we go with the slide one by one changing, sir? Mr. Vivekanandan?
Yes, sir. Yes, sir. I will, sir.
Yeah. You proceed with the next slide. Anaerobic process. Yeah. The acetogenesis, yes, it will convert volatile fatty acid. Mostly, if you see, it is nothing but volatile fatty acid, acetic acid, or propionic acid, citric acid, or valeric acid, particularly. Then this another bacteria, will act on them, converting into methane and carbon dioxide. Now, basically, the microbe process, it is to be carefully handled. Otherwise, it will lead to.
Hello, sir. Hello, sir.
Yeah.
There is a lot of echo. We are not able to hear you properly.
I see. Okay. Why it is an echo is happening? It is not supposed to happen actually. Now it is clear. Hello.
Yes, sir. It's better. Yes. Yeah, it's okay now.
Okay, fine. Now, after this anaerobic process, as such, it is having 4 key factors. One is called hydrolysis. Number 2 is acetogenesis. Third is acetogenesis. Fourth is methanogenesis. Hydrolysis, all the sugars are broken down into all related with the acids like acetic acid or propionic or butyric acid or valeric acid. Next, it is converted into acetates. That is CH3COO. Those things again convert into methanogen bacteria, convert into CO2 and as well as methane. Methane is the key factor for our fuel actually. Main thing is the methanogen bacteria is little bit if you are not maintaining that process pH or the temperature or the sulfate bacteria, if sulfur compound is much more present, what will happen? It will get inhibited by that and your lagoon may fail actually. That's why main key factors are this BFA to alkalinity ratio to be monitored, inlet pH to be monitored, temperature to be monitored. These are the 3 key factors. And we used to have an online monitoring of methane percentage also. So that whatever be the gas it is going inside that lime kiln, where it is replacing the furnace oil, it should have minimum 60 to 65% of methane. And rest will be about CO2 and some moisture and little hydrogen sulfide also will be there. The digestive process begins with, as I was telling you, the 4 processes going to happen. There, methane is the key factor, as I was telling you. We have to maintain a very good equilibrium actually. While doing so, This can be meant out with various equilibriums, like it should not have stressed shock loads. pH should be thoroughly, it should be kept in a uniform rate, and that ratio to be maintained very well. And establishing the equilibrium, as I was telling you, key factor is 25 to 40 degrees centigrade, it should be maintained. pH value should be 6.6 to 7.6 and should not be dropped below 6.2 because methane bacteria cannot function below this level. Sudden fluctuation in pH will inhibit the lagoon performance also. Alkalinity should be in the range of 1000 to 5000 ppm of material should be there. And by continuous monitoring, this ratio should be maintained, and inhibitory substances including ammonia and hydrogen with high concentration of sulfate ions are present, and concentrations such as calcium should be kept to minimum. These are the key factors we have to keep it in mind and to keep the lagoon alive. And I will tell you that ideal conditions, temperature 30 to 35, extreme it can go to 40. Alkalinity 2000 to 3000, extreme it can go up to 5000. Volatile fatty acids 250 to 500, whereas extreme it can go to 2000. Concentration of inhibitory substances which are all detrimental to that operation of that biogas production is sodium should be less than 3500 to 5500. Strongly inhibitory is 8000. Potassium, calcium, again magnesium, sulfide, another critical factor actually. Otherwise, the whole thing will, sulfate bacteria will be acting on that, and your performance of the lagoon will come down actually. The main benefit of advantage of this anaerobic lagoon is it can take care of any shock load also. Instead of digester, this anaerobic lagoon can take care of any shock loads. The on end of this process can be stop, all these things are the shock loads. That's why this lagoon is much more beneficial for us as such. And the methane much more useful. It once it is captured, the whole area corrosion and other aspects have come down for us, and it will produce very less biomass. That is another advantage. Suppose if we are going for this aeration system and all, you may produce much more MLSS. There is a mixture like suspended solids, which is disposal is another issue. And whereas this anaerobic system, biomass generation will be less, and our production cost also very, very less. That is another plus point. Additionally, energy, and we need not aerate it, heat it, or mix it. Less expensive to construct and operate. The biogas generator in the system is very much saving our capex actually. The lime kiln is consuming it for replacing the part of furnace oil, saving a very good extra for the company also. The only disadvantage is it needs a large area of land also for people. Undesirable odor if it is not maintained properly, and relatively longer retention time required when combined to a digester. Wastewater seepage, if it is not maintained properly, the groundwater seepage may likely to happen if the system is lined properly. And environmental conditions directly impact operations, so as any variance limit of the ability to control the process. Any variation is there, we will end up with some issues. The thing is, the main objective, what we learned from the system, madam, everybody, the only thing is the high COD, BOD, which needs to be treated aeration wise, needs a lot of huge power, energy oriented, and under the scenario of renewable energy, we are able to reduce our fossil fuel furnace oil by way of 4 kiloliters per day. That is another benefit for us. And by the way, whatever the money spent on furnace oil is good saving for the company, and the treatment cost will come down in that effluent treatment system. It is universally, it is one of the best paper mill in India using this wastewater for irrigation since 1984 actually. We put up with a very good lift irrigation system. The entire land adjoining our company, all fallow lands in 84 and all sandy loam soil, which is having a very good, leaching property. There will not be built up of salt and all. Every year we are able to average our sugarcane production is 40 to 50 tons. And incidentally, people now changing over to tree crop also, which is much more commercial now, particularly casuarina and as well as eucalyptus. which is another useful raw material for the paper industry. That also people started growing because of the non-availability of the labor also. Because sugarcane crop, it needs some good labor. Even though our sugar industry may get reduction of sugarcane, it is a cycle actually. We ask them to go for an alternative crop also. That casuarina will help them to improve the soil, what do you call it as nitrogen fixation and all. It is much more helpful. And each and every year the productivity is very good. And it is totally a circular economy actually. We used to supply the treated water to the farming community nearby. They will supply sugarcane to the sugar industry. They will take sugar and they will give back bagasse to us. We make paper and again we will treat the water and use it for farming community. This is one closed cycle. And of course, since it is our oldest mill, with all these things, we are able to do a lot of innovative works on reduction in water and utilization of this wastewater for and green belt development within our 3 to 4 kilometer radius. We are able to do a green coverage of the whole area. And by the way, we are able to produce some of the raw material required for us from the nearby sister country in the sugar industry also. This is the closed cycle of our industry. By way of utilizing this biological method and using this treated wastewater for irrigation as well as this for production of biogas, we are much more what do you call it, stronger in pollution prevention in that belt area actually. Thank you, madam. Any more questions? I am ready to answer you. Any queries? Hello. Hello.
Participants, you have any questions?
No issues. Thank you very much.
Dear participants, you have any questions? Sir, I think no questions and all, sir.
Okay, sir.
Dear participants, you have any questions? So there is no question, sir.
Okay, sir. Thank you very much, Sri Venkateshwara College of Engineering and Technology and all. Thank you very much for giving me an opportunity to express our views and our industries. Thank you very much, sir. Once again.
Thank you so much, sir.
Thank you. Thank you.
Thank you, sir. Thank you very much.
Thank you, everybody.
Okay, sir. Dear participants,
Sir, are the sessions for today over?
yes, sir.
Okay,
Sir, I will
so
forward. Yes, sir. I will send the feedback form of session 4, sir. Kindly fill the feedback form. Dear participants, one important thing is yesterday so many people, sir, entered the feedback without registered in the Atal portal. Okay, so many participants are there. For example, 137 members are attended, but the feedback count is around 240. Okay, so without Atal portal entry, if you are attending the workshop with your friend circle support, but there was no use in that, we are unable to generate your attendance and same as well as your certificate in the Atal portal. Okay, so those who are joined without registering in the Atal portal.
Hello.
Yes, sir.
Sir, can you please publish the list of participants, those who are registered with the Atal portal in our WhatsApp group, so that each one can know and register likewise?
Yes, sir. I will update, sir. Nearly 252 members are joined
No,
through Atal
it's a
portal.
huge number. Those who are not yet registered to the Atal portal, you please publish the list of participants, those who have registered on Atal portal, so that we can confirm our name in that list, which you will post on WhatsApp group.
Okay, sir. I will post in the WhatsApp group, sir, list of participants. And then simultaneously, one more important thing, while entering the feedback form, kindly enter your full name, sir. So many persons entering only doctor or Mr., only DR or MR. So we are unable to identify those are attended properly, those are registered in the Atal portal, those are coming from the your friend circle, and all we are unable to identify. So now today's attendance we are updated based on the Atal portal registered candidate attended in the first day of the entire FDP. Shows that what will happen mean, if you are really attended for the entire 4 hours of timing, but you are not registered in the Atal portal, what will happen? I am unable to update your attendance.
Sir, one query I want to ask.
Yes, sir.
I have registered for the course
Okay,
which
sir.
you have scheduled in between 3 to 8th of this month. Then you have rescheduled. So should I register on Atal portal once again?
No, sir. Actually, you are already registered. Means I forwarded the message, sir.
Okay. So we need not to register
Sir,
once
whether
again.
you are
No?
received mail from SVHC Biotechnology Department, sir.
Yes, yes.
Yesterday also
I
I
have
mailed.
received. I
Same
have received the
also
mail.
I mailed.
I have received the mail, sir.
Yeah, no problem, sir. If you received a mail, means you are under the participation list, sir. No problem.
Okay, okay, okay. Thank you, thank
Okay,
you.
sir. Okay, sir. Thank you.
Hello, Mr. Venkat.
Participants kindly. Yes, yes, sir.
Yeah, Venkatji, actually what is happening, if you schedule the class before 6 o'clock, then we are in the office, so we cannot attend. So can you please schedule it after 6:30, because 6 to 6:30 we reach home and then immediately log into the portal.
Sure. Actually, as per Atal FTP, we have to initiate our session at 6 p.m., sir.
Oh,
So
okay,
in between,
okay.
yes, sir. In between that, the session was closed earlier, or otherwise some other is happening. The expert was completed the session early. It means that not at all an issue. But we have to initiate at 6 p.m. in the day 2 onwards. In case of day 1, we have to initiate at 5:30 onwards. At the end, last day of Atal FTP, we have to initiate the session at 2 p.m. That is our schedule, sir. So we have to
Okay.
start from our schedule. We have, if any changes means definitely we will inform. But the timing is sometimes, for example, foreign expert we are requested for foreign expert. The day 6 may be a foreign expert according to the timing. India timing and foreign timing may be changed. It means we can able to modify the timing. Otherwise, if the speaker is in India, we have to follow the as per schedule of 6 p.m. sir. That's why.
Okay, all right. Thank you
Okay,
so
sir.
much.
Okay, participants, kindly switch on your camera. I have to take a screenshot.
Sir, before that, I would like to propose a vote of thanks, sir. Shall I?
Ma'am, just 1 minute, ma'am. After taking photos, we will, ma'am.
Okay, sir.
Okay, ma'am.
Can I have the number of Dr. Shanmugam?
Yes, sir. I will, sir. I think who is asked? I don't know.
Yeah, Lakshmi Prabha, Subha Lakshmi Prabha.
Okay, ma'am. I will, ma'am.
Okay, Subha Lakshmi Prabha. Thank you.
Okay, ma'am. I will send privately, ma'am.
Okay, thank you so much.
Okay, ma'am. In WhatsApp group, my name is Tirunyan Sundam. The mobile number is available, ma'am. 99761. You just send a private message. I will forward the Dr. Shanmugam sir number.
Okay, thank you so much.
Okay.
Thank
Okay.
you.
Okay. Just 1 minute. I have to take screen, sir. Just a minute, sir. Okay, sir. Switch off the camera, sir. No problem. Professor Saladevi, ma'am, you go ahead.
Yes, sir.
Okay, ma'am.
Yes, sir.
You go ahead.
Yes. And now I would like to express my sincere appreciation to our second day resource person, Mr. K. Sanmugam, sir, Senior General Manager, for his vivid and concise presentation. Then I would like to express my gratitude to Coordinator Dr. P. Ramalingam, sir, and Organizer Mr. Tirunyanam Sambandam, sir, for making the second day session possible and ensuring its smooth execution. Thank you, sir. A special thanks goes to our respected faculty members, researchers, students, and all participants for their keen interest and active engagement in today's session. Once again, I thank one and all.
Thank you, ma'am.
Thank
Dear
you, sir.
participants, we are going to signing off today. So tomorrow, the same schedule starts from 6 p.m. I will update the Google link, everything, Google link tomorrow morning. Same time, I will update your attendance in the Atal portal. Thank you. Kindly verify your attendance in the Atal portal. If any modification, if you feel any struggle, kindly make a message through WhatsApp. Okay, and one more important thing, if you are completed your feedback, don't acknowledge in our WhatsApp group, because we are giving any messages mean continuously you are given the acknowledgement like submitted and some screenshots are available means my message will reach very late. That's why if you are submitted means no issues, everything is a recordable one, so your attendance, it will be recorded automatically. So you just fill the feedback, that's enough. If you are not filling means personally you just text me, I will help you. I think you maybe understand the situation in WhatsApp group, same as well as in the attendance. So kindly fill your feedback form with the proper name. Okay, very few of the persons are continuously rejoining. I think you are attending in mobile phone. So what will happen means, once if you are entered the Atal FTP means it's automatically recordable. So once you are left during your call or message or anything, if you are left from this Atal session, what will happen? Automatically your attendance also reduced. Then if you are rejoined means it's considered as a, once again considered as a starting from first minute. So what will happen? Your attendance ratio is continuously reducing. So try to understand the situation. Your attendance is very much important. Up to 80% of attendance only able to get the certificate. So we are just organizing the FDP, the entire certificate and marks, everything is in the Atal portal only. So based on your final test and your feedback and then your proper attendance of day 1 to 6 with minimum 80% only to possibilities to get the certificate. So kindly understand the situation and react the same. Thank you so much for your contribution. Tomorrow we will meet again. I will send all the details in the WhatsApp group. Thank you so much, participants.
Yes, all the participants are asked to join for day 3 session tomorrow at 6 p.m. sharp. Hope you will be punctual in joining us tomorrow. Thank you. Good night.